Bismillahirrahmanirrahim. Assalamu alaikum dear students. Today I'm here with the lecture number 30, that is primary, secondary and tertiary structure of proteins. The contents of today's discussion are first of all a brief introduction about protein and their classification, and then we will move towards function of these proteins and the structure of proteins and the classification on the basis of the structure of these proteins, and then we will see in detail first of all primary structure and then secondary, and finally we will discuss tertiary structures, and then a few references mentioned at the end of this lecture and a few assignments. So let's start with proteins and their structure. So proteins are the most abundant organic molecules of the living organisms. More than 80% of the biomolecules, they are basically proteins. So our body is mainly made up of organic molecules that are mostly proteins. So they constitute about 50% of the cellular dry weight. So you can imagine the constituent of the human body, that 50% of the cellular dry weight is made up of these proteins. They constitute the fundamental basis of structure and function of life. In 1839, Dutch chemist named as G. de Mulder was the first who described about the proteins by name. The term protein is derived from a Greek word that is proteos, and it means first place, right? So the thing that is required in first place or is of top priority is basically called as proteos. The proteins are nitrogenous macromolecules that are composed of many amino acids. What are amino acids? Amino acids are a group of organic compounds containing two functional groups, that is, one is amino and the other is carboxylic group. The amino group, that is NH2, is basic, while the carboxylic group, that is COOH, is acidic in nature. There are about 300 amino acids that occur in nature, but only 20 of them occurs in proteins. So only 20 different combination of these 20 out of 300 makes proteins. So here is the general structure of amino acid. Here you see is the amino group, and here is the carboxylic group, and here is the R group, and this is basically alpha carbon with which amino and carboxylic as well as the R groups are attached. Structure of amino acid become about it that each amino acid has 4 different groups attached to alpha carbon, which is carbon next to carboxylic group. So alpha carbon is basically carbon that is next to carboxylic group. These 4 groups are amino group, carboxylic group, hydrogen, and R, that is a side chain that is different in different amino acids. So here you see the protein and the different amino acids: glycine, alanine, valine, leucine, methionine, isoleucine. So there are 20 amino acids that make these proteins, and their structure is mentioned here. Some of these are non-polar. So here you see all of these are non-polar with aliphatic R groups. All of the groups that are R groups are aliphatic in nature. You see here some are polar and uncharged R group. So these here you see these are uncharged R groups, and these are basically polar. And some are positively charged R groups. Here you see lysine, arginine, histidine, they are basically charged R groups. So some are negatively charged R groups. So you see here aspartate and glutamate, they are negatively charged, while some are non-polar aromatic R groups. So here you see phenylalanine, tyrosine, and tryptophan, they are they are having aromatic R groups. So what is the function of these proteins? Proteins also receive signals from outside the cell and mobilize intracellular response. They are also the workhouse of macromolecules of the cell and are as diverse as the functions they serve. So as their functions, there are a lot of functions, there are many functions they perform, so their structures are also as vast as or as diverse as their function. In our previous lecture, we discussed all of the functions of these proteins. So the difference between the globular and fibrous protein is we already discussed in our last lecture. So their shape is different. Fibrous are long, while globular are round and spherical. The purpose of the fibrous is structural, while globular is functional. Acidic sequence, repetitive amino acid sequence, by in globular form you have irregular amino acid sequence with durability. Then fibrous proteins, they are less sensitive to change in pH and temperature, etc. While globular, they are more sensitive to these conditions. And examples of fibrous is collagen, myosin, fibrin, actin, keratin, and elastin. Examples of globular protein is enzymes, hemoglobin, insulin, and immunoglobulin. When it comes to solubility, fibrous proteins are basically insoluble in water, while globular proteins, they are soluble in water. Then here are some amino acids. They can be non-polar, they can be polar, they can be acidic, they can be basic. If we have already discussed in the previous slide about non-polar, polar, and acidic and basic. Basically, non-polar amino acids, they are hydrophobic, and examples are adenine, valine, leucine, and here is a structure and example, that is the leucine. And polar are hydrophilic, non-charged. Glycine, serine, threonine are example, is the structure of threonine. And some are acidic. If they have extra carboxylic group, they will be acidic and they will be negatively charged. And example is aspartic acid. And here is the structure. And this, if they are basic, they are positively charged. Lysine, arginine, and histidine is example. And here you see the structure of lysine. And it is having positive charge. Now, how peptide bond is formed? Alpha carboxylic group of one amino acid with the side chain R1 forms a covalent peptide bond with alpha amino group of another amino acid by removal of a water molecule. So this results in the formation of a dipeptide because two of the amino acids are get together. So this is dipeptide. The dipeptide can then form a second peptide bond with a third amino acid that will give rise to tripeptide, and this way we get the polypeptide chain. The repetition of this process generates a polypeptide protein of specific amino acid sequence and has 40% double bond character. These polypeptide chains that caused by the resonance. So here you see the formation of a peptide bond. There is one amino acid, here is a second amino acid. What happens that amino group of one amino acid reacts with carboxylic group of other amino acid and water molecule H2 and O from here. Water is removed and there is a bond formation, that is amide bond, that is called peptide in case of proteins. Right? Basically it is an amide, but in proteins we call it as peptide bond. So here you get the dipeptide. Polypeptide bond is the repeating sequence of basically NCC, NCC. So this is in the peptide bond. The side chain or the R groups is not a part of backbone or the peptide group. So they are basically the side chains. They are considered as side chains because they do not participate in the formation of backbone. Backbone are just carbonyl and amino group and alpha carbon. So here is an overview of protein structure. According to configuration, geometric relationship between a given set of atoms example that distinguish L amino acids from D amino acids. So by configuration we can say that amino acids can be D or L. So it comes conformation, that is the spatial rearrangement of atom in the protein. Then thermodynamically the most stable conformation exists and stabilized largely by weak interactions. And this stability is basically tendency to maintain the native conformation or the parent conformation, that is called the stability of protein. And what is native conformation and how it is stabilized, as already I told you that there are many forces that are basically not true bondings, so they are basically responsible for its native conformation stability. That are basically disulfide bonds and non-covalent forces. Now, when it comes to structures of proteins, proteins have different levels of structure or organization. The first one is primary structure, the second one is secondary and tertiary, and then quaternary structure. Among these four, primary is the simplest organization level, and then is the next complex one is secondary, and tertiary is more complex as compared to secondary, and the most complex level of organization of protein is quaternary structure. So here you see the primary is just a combination of different amino acids through peptide bond, and then these become in folded form. You see this is secondary structure. They can be in alpha helix form or they can be in beta sheet form. Secondary basically exists in these two forms, and then from secondary structure we get the tertiary structures. They are a little bit more complex. These are basically three-dimensional structure of proteins. Then these tertiary structures become more complex and they form quaternary structure. So what forces determine the structure of proteins? So primary structure or the primary proteins, basically they are basically involve only covalent bonds, so their forces is determined by covalent bonds only, while the secondary tertiary quaternary structure they are involve many weak forces. So here you see the primary structure. This is a polypeptide chain of amino acids, and they are basically assemblies. And the secondary structure, they can be in plated form or they can be in helix form. So they are basically foldings of these primary structures. So the tertiary structure can be combination of both plated sheets and alpha helix. So you see here the alpha helix, and this is the plated form. So these tertiary structures are can be combination of these two, and these are basically packing of the proteins. And then these basically tertiary structures, different tertiary structures, they combine together to give quaternary structure, and these are basically interactions between tertiary structures. So let's start with primary structure. The primary structure of protein refers to the sequence of amino acids present in the polypeptide chain. Amino acids are covalently linked by peptide bonds or the covalent bond. Basically, peptide bonds are covalent bonds. So each component amino acid in a polypeptide is called a residue or a moiety, right? So a monomer is called a residue or a moiety. By convention, the primary structure of protein starts from the amino terminal and ends at carbon terminal. So primary structure have basically terminals. One is amino terminal and the other one is carboxyl terminal. So one is negatively charged, one end is, and the second end is positively charged. Overall, it is neutral. And then why there are many important things that are related with primary structure. Number one is they are very important to predict secondary and tertiary structure from the sequence homologies with related proteins, because secondary and tertiary structures, they are made up of primary structure. So if we know the structure of primary protein, then we can move towards the structure prediction of secondary and tertiary proteins. And many genetic diseases, they are because of the result of abnormal amino acid sequence, that is basically in primary structure of the protein. So whenever we need to study these abnormalities, we have to find out these genetic abnormalities. We have to study the primary structure of these proteins, and they can be used to understand molecular mechanism of action in proteins. So what type of mechanism of protein, because proteins are involved in many reactions in the body, and there is a specific mechanism with which they perform. So if we want to know this mechanism, we want to understand this mechanism, what we need, we should know the structure of these proteins, how they are made up of. Then we will come to know how they are performing their function, what mechanism is involved in their function. So they can also be used to trace evolutionary paths, because evolution is basically change in genetics. So what change in genetics of proteins, because we are basically made up of proteins. So evolution is basically change in primary structure of the proteins. So we can trace evolutionary paths by the study of primary structure of proteins. Now comes the secondary structure. It is a local, regularly occurring structure in proteins and is mainly formed through hydrogen bonds between backbone atoms. Right? Pauling and Curie studied the secondary structure and proposed two confirmations. So he finally find out that the secondary structures, they exist in two forms. Number one is alpha helix, helical form, and number two is beta sheet, that is the sheet form. So here you see the two forms of the secondary structure. One is helical, you see a helix, and this is plain. This is the sheet form. They exist in basically these sheet forms. Okay. So what is alpha helix? This is basically coiling of protein chain or the amino acids in shape of a right-handed helix, that is alpha keratin. So the important point is here is right-handed. It can be left-handed, but proteins in the biological body, they exist in right-handed helix. I will tell you in the next coming slides what is right-handed helix. So here you see these are the basically coilings, right? Coilings or the foldings. So this is alpha helix. Carbonyl oxygen of an amino acid residue is hydrogen bonded to the amino hydrogen of the fourth amino acid residue further down polypeptide chain. So here you see the carbonyl of one is making hydrogen bond with the fourth amino acid, amine of fourth amino acid. You see here the first and the fourth. One. So this is 1, this is 2, this is 3, and this is 4. So it's the formation of, this is the first carbonyl, right? First amino acid carbonyl. This is the first amino acid, this is the second amino acid, this is the third amino acid, and this is the fourth amino acid. So the carbonyl of first amino acid is making hydrogen bond with the amino of fourth amino acid. And the same pattern is repeated throughout the alpha helix, right? So amino acid per turn are about 3.6, means around 4. 4 of the amino acid make one turn. So this is what is right-handed alpha helix. So this is right, means if you put a thumb here, then the folding is going on the right side, right? So this is right-handed alpha helix, and if this folding going on the left side, then it means it is left-handed helix. So naturally or you can say in biological system, they exist only in right-handed form. And whenever the protein or the secondary protein is in right-handed form, it can perform biological function. But if it is the same protein is in left-handed helix form, it cannot perform biological function. That is very important thing. So this is a alpha helix. You see a hydrogen bonding between hydrogen of amino group and oxygen of carbonyl group is taking place, and this is a helix, right? It is moving like a helix. So carbonyl oxygen of an amino acid residue is hydrogen bonded to NH hydrogen of the fourth amino acid residue further down the polypeptide chain. So what is beta pleated sheets? You see they are in the form of sheets. This is a sheet, folded sheet. So a side-by-side alignment of adjacent polypeptide chain. So you see here the first chain and the second chain. So they are moving each other side by side, one side and the other side. So they are making a sheet. So individual polypeptide beta strand. This is individual strand, right? Each beta strand is fully extended, right? It is fully extended. They are stabilized by hydrogen bond between NH and carbonyl groups of adjacent chain. You can see here in this figure that there is a hydrogen bond formation between NH of one sheet and carbonyl of the other sheet. And the same is here. Amine of hydrogen of one and carbonyl oxygen of other. They are making hydrogen bonds. So the sheet conformation permits the stacking of sheets one on the top of the other. Beta sheets come in two varieties. They can be anti-parallel beta sheets or they can be parallel beta sheets. Anti-parallel beta sheets are neighboring hydrogen bonded polypeptide chain run in opposite direction. While in parallel beta sheet, hydrogen bond chains extend in the same direction. So you see here, this is one of the beta sheet, and this is the they are running in the opposite direction. Okay. This is parallel and this is anti-parallel. Oh, sorry, this is anti-parallel. See here, it is moving this way and it's moving this way, and these are basically parallel. They are moving in the same direction. So the connection between the two anti-parallel strand may be just a small loop. So there is a very small loop. You see here, loop is very small. This is a small loop. This is also a small loop. But the link between tandem parallel strand must be a crossover. So you see there is a crossover interaction, means from here it is making cross like this and another cross, right? That is out of the plane of the beta sheet. It is not in the plane. This is the plane and this is out of plane. So here you see two types of beta plated sheets. This is parallel and this is anti-parallel. You see here this is the N-terminal, means they are moving in the same direction. You can see here you have N-terminal and N-terminal of both sheets at the same side, and C-terminal and the C-terminal on the same side. But in anti-parallel sheets, these two terminals are in opposite direction. Means one of the sheet is having N-terminal while the other of the sheet is having C-terminal in front of each other. Same is here. At the other side, one is having C-terminal and the other is having N-terminal. So this is parallel when they are having same terminal at the same side, and they may be anti-parallel if they have opposite terminals on the same side. So two major sorts of connections between beta strand takes place: a hairpin or same end connection, a crossover, or the opposite end connection. So you see the hairpin or the same connection exists in parallel sheets, while the crossover or opposite end connection occurs in anti-parallel sheets. So you see here, it can be like this. From the down, it can have, or it can have from the up. So it can be right-handed or left-handed in case of anti-parallel, or in case of parallel beta sheets, it will be like hairpin. You see, it's the shape of a hairpin. What is dihedral angle or the torsional angle? That is the angle between two intersecting planes. It helps to maintain the protein structure. So this is basically the torsional angle between the two beta sheets, right? It's very important to maintain the structure of the two proteins. So here you see there are two types of angle. One is called psi and the other one is phi. So the polypeptide chain conformations, the backbone or the main chain of a protein refers to the atoms that participate in peptide bond, ignoring the side chains of the amino acid sequences. The only reasonable free movement are rotation around alpha carbon and nitrogen bond, that is measured as phi, and that is measured as phi, and alpha carbon and the carbon bond, that is basically you can say carbonyl carbon, that is measured as psi. So there are two different angles that are the psi and the phi that are important, because rotation around these is possible, otherwise it is a rigid structure. So these angles are both defined as 180 degrees when the polypeptide chain is in full conformation. So here you see this is the phi rotation and this is the psi rotation. Between carbonyl and alpha carbon it is psi, and between alpha carbon and nitrogen it is phi. The conformation of the backbone can therefore be described by the torsional angle, also called dihedral angles or rotational angles. The peptide bond allowed for rotation around it, and therefore the protein can fold and orient R groups in favorable positions to minimize the steric hindrance. The weak non-covalent interaction will hold the protein in its functional shape. These are weak and will take many to hold the shape. So there are a number of weak forces, because weak, when they are in large number, they become strong. So that is the reason these are many in numbers. The protein foldings occur in the cytosol. Here you see the protein, that is the primary structure of proteins, how they become in the folded form. To hold this folded form, there are a number of interactions. Mainly they are hydrogen bond interactions between oxygen of carbonyl and hydrogen of amino groups. So these many hydrogen bond maintain the structure of the secondary proteins. Second regular folding pattern have been identified formed between the bonds of the peptide backbone. Alpha helix protein turn like a spiral fibrous proteins, that is basically present in hair, nail or horns. So this is another form, that is alpha helix. Right. So it exists in helical form. So it is alpha helix. And here also there are hydrogen bonds. You see the number of hydrogen bonds between oxygen of carbonyl and nitrogen of the hydrogen of the amino. So here is the beta sheets. Proteins folds back into itself in a ribbon or globular form. You see there are also hydrogen bonds. So turns and loops. In addition to alpha helix and beta strands, a folded polypeptide chain contains two other types of secondary structure that are called loops and turns. Loops and turns connect alpha helix and beta sheets, right? So basically, how alpha helix and beta strand, that are two types of secondary structure, they are connected with each other through loops and turns. The most common type of cause a change in direction of the polypeptide chains, allowing that allow it to fold back on itself to create a more compact structure, right? So these loops and turns are basically used to make the secondary structure more complex or compact. Loops have hydrophilic residues and they are found on the surface of the proteins. Loops that have only 4 or 5 amino acid residues are called turns when they have internal hydrogen bonds, right? So when they have only 4 to 5 amino acids, the loops are called turns. Reverse turns are a form of tight turn where the polypeptide chain make a 180-degree change in direction. So these are reverse turns. Reverse turns are also called beta turns because they usually connect adjacent beta strand in beta sheets. So here are the turns and the loops. This is basically a loop and this is basically a turn. So what a beta turn, also known as beta bend or tight turns. In a beta turn, a tight loop is formed when the carbonyl oxygen of one residue forms a hydrogen bond with the amide proton of an amino acid 3 residue down the chain. So it is in between the first and the third residue. This hydrogen bond stabilizes the beta bend structure. Proline and glycine are frequently found in beta turns. Proline because its cyclic structure is ideally suited for the beta turn. The glycine because with the smallest side chain of all amino acids, it is most strictly flexible, sterically flexible. So because there are very small groups attached with C carbon, so this is sterically flexible, and proline is basically as told here that it is cyclic structure. That is why they are suitable for beta turns. So at beta turns, generally proline and glycine are present. A beta turn is a means by which the protein can reverse the direction of its peptide chain. Beta turns often promote the formation of anti-parallel beta sheets. So you see with the beta turn, that is beta band, permit the change in the direction of peptide chain to get folded structure. So this is basically a beta turn. You see a glycine part, a very simple amino acid. It gives protein globularity rather than linearity. So to make proteins more globular, there are more beta turns. Then comes the tertiary structure of the proteins. The tertiary structures define the specific overall 3D shape of the protein. Tertiary structure is based on various types of interaction between the side chain of the polypeptide. So basically, the interaction between the side chains is responsible for tertiary structure, because in the secondary structure we see that there was involvement of only hydrogen bonding between the two amino acid residues. But here, in tertiary structure, there are many other interactions between the side chains. Okay, so here you can see a number of, if this is alpha helix or helical form, it can be beta plated form. You see, it is a plated form, and there are hydrogen bondings between the beta plates. There are disulfide bonds, there are salt bridges, there are hydrogen bonding, there are hydrophobic interactions, there are hydrophilic interactions to water. So all these forces together make the tertiary structure of proteins stable. Tertiary structure are three-dimensional structures that are generated by interaction between the amino acid residue of functional proteins. So, there are two types of tertiary proteins: globular and fibrous. So, in globular proteins, the tertiary interactions are frequently stabilized by sequestration of hydrophobic amino acid residues in the protein core. The consequent enrichment of charge or hydrophilic residue on the protein's water exposed surface. So basically, globular proteins, the hydrophilic parts are outside, on the outer part of the protein. In secreted proteins, the disulfide bonds between cysteine residue help to maintain the protein tertiary structure. So these disulfide bonds basically play an important role. Interactions that stabilize tertiary structure are disulfide bonds, hydrophobic interactions, hydrogen bonds, ionic interaction, van der Waals forces, and hydrophilic interactions. So here you see the tertiary structure, disulfide bonds, when they are formed. As I told you, the cysteine residues are important because they have SH group attached. So there is a disulfide bond formation between them. So between cis and the cysteine and cysteine, there is a formation of disulfide bond that make these tertiary structures stable. So two of the hydrogen are removed, or hydrogen molecule is removed to give this disulfide bridge. Hydrogen bonds are weak, which allow to be broken and reform easily. So these are basically hydrogen bonds that are weak, but there are a lot of hydrogen bonds in tertiary structure. So many of the hydrogen bonds are present that becomes strong. So they allow structural changes and produces functional molecules. Here you see between oxygen of carbonyl and hydrogen of amine, there are hydrogen bonds. And there are some salt bridges also. Salt bridges are electrostatic bonds between oppositely charged groups, negative and positive charged groups. And the strength is usually 4 to 7 kilocalories per mole. So these are, you can see here, the salt bridges between the protonated amine and you can say negatively charged carboxylate part. So these are basically salt bridges, that is the electrostatic bond between the oppositely charged species. So here are the different stabilizing forces and their examples: hydrophobic interaction, and the nature of bonding is attraction between non-polar alkyl and aromatic groups from non-polar center, that is repelled by water. So examples are basically methyl groups, and methanol, you can say, or CH2OH, and water. So these are hydrophobic interactions. And hydrophilic interactions basically are attraction between the polar. This is the example of hydrophobic between the two methyl groups, and this is the example of hydrophilic, that is methanol, that is positive, that is charged species, partial charge. This also has partial charge, oxygen has partial negative charge, and here hydrogen has partial positive charge. This is an example of hydrophilic interaction. So attraction between polar or ionized R groups and water on the surface of the tertiary structure. Then are the salt bridges, we already discussed it. Hydrogen bonds, already discussed it, and disulfides have also been discussed earlier. So the globular proteins, when it comes, they are basically form of tertiary structure. The globular protein fold up into compact spherical shape. Their function include biosynthesis, transport, metabolism. For example, myoglobin is one of the globular protein that store oxygen in the muscle, and myoglobin is a single peptide chain. You see here, a single peptide chain that is folded, right? that is mostly in alpha helix form. The oxygen binding pocket is formed by heme group. Right? See, you see here there is a pocket formation where you can see a heme group and specific amino acid side chains that are brought into specific position by tertiary structure. So then another form of these tertiary structure, that is fibrous protein. Fibrous protein consists of long fibers. You see here the long fibers. These are basically long fibers and are mainly structural proteins. For example, keratin are fibrous proteins that make hair, fur, nail, and skin. Hair is made of twinned fibrils, which are braids of three alpha helical, 1, 2, and 3. 3 alpha helix, similar to triple helix structure of collagen. The alpha helix are held together by disulfide bonds, and beta carotenes are fibrous proteins found in the feathers and scales that are made up of mostly beta plated sheets, right? So alpha carotenes, they are made up of alpha helix, and beta carotenes, they are made up of beta plated sheets. Then are the myoglobins. Myoglobin is a small monomeric protein which serves as an intracellular oxygen storage site. It is found in abundance in the skeletal muscle of vertebrate and is responsible for the characteristic red color of the muscle tissues. Myoglobin is closely related to hemoglobin, which consists of 4 myoglobin-like subunits that form a tetramer and are responsible for carrying oxygen in the blood. Myoglobin we already discussed in our previous lecture, very detailed discussion. So we don't need here. Just want to tell that this is myoglobin is basically made up of beta turns. You can see here alpha helix and beta turns. This is beta turn. You can see an alpha helix mainly. So after this detailed discussion about primary, secondary, and tertiary structure, I hope and I'm sure that all of you know what is primary, secondary, and tertiary structure. So now you have to find out the significance of each class of the compound. You have to find out number one, biological significance of primary proteins, then for secondary amine, and then for tertiary. At least one significant rule in biological system, and then you have to find out what are the stabilizing forces for these structures, meaning which forces or which bonds stabilize primary structure and which forces are stabilizing secondary structure and which forces are stabilizing tertiary structure. You have to write down all these on a piece of paper, right? So here are a few references for your convenience. If you want to go and consult them, you will find these proteins structure, that is primary, secondary, and tertiary structure in detail. So for today, that's enough. So take care and enough.